Hello everybody, welcome to the last chapter in HAPch15part1, which is all about the autonomic nervous system. Um, we have gone through already a long journey through the nervous system. We looked at the basics of how neurons work. We have looked at gross anatomical structures and wiring diagrams for the physiology of the spinal cord. And we have looked at tens of thousands, or seems like tens of thousands of parts inside our brain. We learned about those four parts, and even within those four parts, we looked at smaller gross anatomical structures that fulfill highly specialized functions. We have put an emphasis on activities that is relatable to our conscious experience. Here in our autonomic nervous system, we will be focusing on events in the body and their regulation by the nervous system that is beyond our conscious control. When you look at this very first slide, we are talking about regulation of smooth muscle, cardiac muscle, and gland. If you look way back to the tissue chapter or in the muscle chapter, chapter 10, you remember that smooth muscle and cardiac muscle are involuntary. We have no conscious control over them. Glands, we have absolutely no conscious control over the activity of these portions in our body. So we are talking about a part of our nervous system that has thousands of reflex arcs hardwired into it. This comes with us with our birth. During embryonic and fetal development, all the gross and microscopic anatomical structures of this automated part of our nervous system are built. And the end result is maintaining homeostasis by regulating a number of smooth muscle structures in the body, cardiac muscle, and glands. The reflex arcs have a huge variety of beginning. So autonomic nervous system monitors, they have receptors in many body parts. But in addition to those, limbic system and some regions of the cerebrum can also provide input to the working of the autonomic nervous system. So when you say reflex arcs from chapter 13, I hope your mind immediately jumps into this imagery of seeing sensory neurons and integrating center, motor neurons, and effectors. So the sensory neurons are mostly what we would call interoceptors. They are inside our body. They are not exposed very much to the outside environment, with a few exceptions. The flow of nerve impulses from these sensory neurons are not consciously perceived. This means that the sensory nerve impulses from our autonomic receptors are not going to the cerebrum. They go to other parts of the brain for integrating function. And here are those some other parts of the brain: hypothalamus, this is part of the diencephalon, brainstem, and spinal cord. On the motor portion of this reflex arcs, once a decision is made by these integrating structures, nerve impulses will stream through motor neurons to the effectors, the structures in a body that's supposed to carry out a function to maintain homeostasis. And this would be cardiac muscle of the heart, smooth muscle in a huge number of organs, and glands. So we are instructing through our autonomic nervous system the functioning of cardiac muscle, smooth muscle, and glands. So let me give you an example that will give you an idea about the wide variety of sensory neurons that will include integrating center either in the hypothalamus, brainstem, or spinal cord, and will include the effectors, cardiac muscle, smooth muscle, or glands. So let's take a look at regulating body temperature. Our core body temperature is set at 100 degrees Fahrenheit. It is a homeostatic measure. We are actively regulating it, maintaining it in a normal range. The sensory neurons are thermoreceptors in our skin and inside our hypothalamus. Our hypothalamus monitors actively 24/7, even when we are sleeping, the temperature of the blood that is flowing by it. So that's the sensory portion. Integration is happening inside the hypothalamus, and whether we are hypothermic or hyperthermic, going either below or above the normal core body temperature range, the hypothalamus will instruct effectors. Well, let's talk about hyperthermia. Maybe in AMP2 we have a little more detailed discussion of core temperature maintenance. On a summer day, heat index 110, you go outside and your body is starting to heat up. Your sensory neurons will detect this heating up of the blood. The hypothalamus will send nerve impulses, totally automated fashion, to, among other structures, glands. You remember those eccrine sweat glands in the skin? Well, they receive nerve impulses from the hypothalamus, and those nerve impulses will tell the eccrine sweat glands secrete sweat. Hopefully evaporative cooling will return core body temperature to normal, but we have just described a reflex arc maintaining body temperature and where nerve impulses are flowing through the autonomic nervous system. Let's take a look at another example, automated adjustments of the workings of the body. Let's say you go out for a 5-mile run. At the very beginning, you have a heart rate of 75, but about half a mile into this run, your heart rate is up to 100, and later on may go up to 120, depending on how fast you're running. What is happening in a body? This is an automated adjustment to maintain carbon dioxide and oxygen homeostasis in the body. We have no conscious control over it. Sensory neurons detect buildup of carbon dioxide in your blood. Sensory input is going into the brainstem. And in a brainstem, we have a cardiovascular center, and that cardiovascular center receiving nerve impulses from the chemoreceptors will tell the heart to increase heart rate. So now we use the brainstem as an integrating center, a different set of sensory neurons, and now we adjusted the workings of cardiac muscle. Let's take a look at another scenario. It happens quite a few times a day, but maybe it's more relatable for infants and toddlers before their potty training days. If you raise kids, you must have changed quite a few diapers. I surely did my share. So what is happening in the infants and toddlers before potty training? Can't they just hold their pee and poop and wait until we put them on a toilet? Well, they can't, because their defecation and urination, which we'll be calling in AMP2 the micturition reflex, is integrated by the autonomic nervous system at those early months of our lives. What's happening in those reflexes? Let's talk about urination. In our bladder, we have stretch receptors, and the filling of the bladder is detected by the sensory neurons. The nerve impulse will go to our spinal cord, And our spinal cord is going to send nerve impulses to smooth muscle of our urinary bladder. Those smooth muscles jump into action. They squeeze on the urine. The sphincter that closed the bladder will be relaxed by those nerve impulses, and urination happens. And time to change another diaper. The way we achieve potty training is to utilize skeletal muscles that form a sphincter around the ureter, around the urethra, and that's how we learn to control and postpone urination. Without that potty training, without that skeletal muscle, everything is on autopilot. So these are just 3 examples showing automated workings of our body. Again, these are based on reflex arcs that have the traditional parts to them. The integrating aspects are located in the non-consciously working part of our brain. So this is our justification for looking at in the details of the autonomic nervous system, maintaining homeostasis 24/7 without our conscious intervention. This is a humongous achievement. Think about human development. We start from a single cell, yet somehow in those 46 chromosomes of our DNA, there is the master plan to build this kind of part of the nervous system. It's just tremendously humbling what goes on in those first 2-3 months of our embryonic development. Beginnings of our autonomic nervous system starts at that point. In AMP2, in the cardiovascular, respiratory, urinary, digestive, reproductive systems, we will come back to specific examples. Here in chapter 15, we're just giving the general layout, which means a generous amount of gross anatomy. In the second part of this lecture, we'll look at a little bit of microscopic anatomy, and we'll be looking at neurotransmitters and some other aspects of the autonomic nervous system. In the second part of this lecture, we will also look at a more everyday occurrence of overarching regulation of our autonomic nervous system, and this is going to be the introduction of rest and digest phase of our life, or fight or flight response. Those two are coming to our body courtesy of our autonomic nervous system. So let's take a look at autonomic nervous system as opposed to the somatic one that is based on our conscious awareness, and we have conscious control over that. So we're going to have differences how the motor neurons are carrying nerve impulses in the last part of the reflex arc, and we will have gross anatomical differences coming from this. So if you want to send a nerve impulse to skeletal muscle, we need a single neuron going from a spinal cord to that skeletal muscle. We mentioned in chapter 10, we release acetylcholine as a neurotransmitter to instruct skeletal muscles to start contracting. In the autonomic nervous system, if you want to send a nerve impulse to a smooth muscle of the bladder, the cardiac muscle of the heart, or that eccrine sweat glands, that the examples that I mentioned, we need 2 neurons. And don't ask why. This is just how the autonomic nervous system is built for the motor portion. Again, this is only the motor portion, and the neuron does start out from a spinal cord, but then it comes to a stop, and a switching of the nerve impulses must happen. As you might imagine, synaptic transmission of a nerve impulse occurs here. These switching points are composed of millions, sometimes tens of millions of cell bodies, in dendrites and axon endings. So these are gross anatomically visible structures. These are called the autonomic ganglia in plural. So we will talk about pre- and post-ganglionic neurons here in this autonomic nervous system. For the motor portion, it is important to recognize that new gross anatomical structures are created in the body just because of this basic design of the autonomic nervous system. So preganglionic neuron, it's pretty normal, what you'd expect in our nervous system, insulated or type B fiber. The postganglionic neuron that will contact the target cells have no myelination. So this is the, what I'm aware of, the only example in the human body where by design we have no insulation of axons. And again, don't ask me why. This is just how this part of our body is built. So on that basic motor aspect of our body, we also have two ways to deliver nerve impulses to a single organ. And again, this is all about the motor portion of that reflex arc. We will not coming back to sensory receptors or the integrating portion. To deliver nerve impulses, instructions to cardiac muscle, smooth muscle gland, there is a level of sophistication in our autonomic nervous system that allows the nerve impulses to speed up or slow down a particular organ. The speeding up or slowing down nerve impulses are coming through two different bundles of axons, and so we have a gross anatomically separate sympathetic and parasympathetic division to the autonomic nervous system. This will leads to dual innervation of many organs in the body. This is a very unique characteristic of the autonomic nervous system. So let's take a look at this image, and here you see the two divisions. The parasympathetic division is where you see the motor neurons in blue, and please keep remembering this is only the motor portion. That's the only thing that we'll be focusing on here. In AMP2 we'll come back to the sensory receptors and the integrating centers. Here we're just emphasizing the anatomical differences and characteristics of the motor portion. So let's take a look at the parasympathetic division. Here you have the first neuron, the presynaptic neuron of the motor division, and you see these switching points forming a U-shaped set of structures all the way to the sacral portion of the spinal cord. These are gross anatomically visible structures. Every little bump here that you see has the size of a black pepper or maybe a little larger. So these are visible to the naked eye. These are called a sympathetic chain ganglia. So this is the sympathetic division in blue. The postsynaptic neuron goes all the way to the target organ. The sympathetic chain ganglia are very close to the vertebral column. So the first neuron from the spinal cord to the chain ganglia are very, very short. The second neuron, the postsynaptic neuron, is very long. Also notice the exit points of the sympathetic division bundles of axons only from the thoracic and the lumbar portion of our spinal cord. So the sympathetic division is also called thoracolumbar for that reason. And don't ask me why it's not coming out from any other part of the spinal cord. Let's take a look at the parasympathetic division. The parasympathetic division neurons are labeled in red here. And take a look at the exit point from the central nervous system, either the brain or the sacral division of our spinal cord. So this parasympathetic division is sometimes called craniosacral because of the gross anatomy. And as you can look, directly compare parasympathetic with the sympathetic side, the exit points from our spinal cord or brain are mutually exclusive. No parasympathetic or red neurons are leaving spinal cord at these levels, and same is true here. No sympathetic neurons are leaving the brain or spinal cord at these levels. So these are constructed gross anatomically totally separate ways. We will be also going into the detail of some of these pathways, some of the ganglions, and this is where a lot of unusual names will come up. This is also a good place to illustrate dual innervation. If you take a closer look at the target organs, we have, for example, lacrimal glands, salivary glands, we have the heart, bronchioles inside the lung, the liver, the stomach, the pancreas, small intestine, large intestine. Look at all these blue neurons carrying sympathetic nerve impulses to that. Well, we have parasympathetic nerve impulses going to the same organs. So a salivary gland is dually innervated because it is getting both a sympathetic and a parasympathetic set of nerve impulses. Same is true for the heart, same is true for the bronchioles of the lung, same is true for all organs of our digestive system here in the abdominal cavity. So this is the craziness of the autonomic nervous system. We are sending 2 bundles of axons to all these organs that participate in dual innervation. In terms of the sympathetic division, we have to mention the sympathetic chain ganglia, and we also have ganglia in the abdominopelvic cavity. These are the prevertebral ganglia. When it comes to the parasympathetic structures, besides the difference that I mentioned, the cranial and spinal cord origin of the preganglionic cells, the postganglionic cells are very short. They may be 1/4 of an inch long or sometimes microscopic because the ganglia are sitting inside the walls of the organs or inside the organs themselves. So this is the illustration of the length of the preganglionic and postganglionic neurons. Here's the sympathetic division in blue, short preganglionic, long postganglionic neuron. And here for the parasympathetic, there's a long preganglionic and a very short postganglionic neuron. Right here in the middle, there's something unusual. The sympathetic division of our autonomic nervous system sends nerve impulses to an endocrine gland. This is an unusual situation. Usually exocrine glands are regulated by the autonomic nervous system, but here the adrenal gland and the adrenal medulla is also getting nerve impulses. The adrenal medulla will be secreting neurotransmitters into the blood, and we'll see the effect of all that in the second part of this lecture. So we are describing the basic neuronal pathways circuitry of the autonomic nervous system. This is just this slide is just to illustrate plexuses in the context of the spinal cord. We mentioned the plexuses of spinal nerves. Well, autonomic nerves also form meshworks, and so if you take a look at the size of the sympathetic chain ganglia, you see these yellow bumps here. Maybe it's best to compare the size to the aortic arch. The aortic arch has the width of your pointing and middle finger, so this is 2 finger wide. And when you look at the size of the chain ganglia here, you can see that they are much smaller. Again, I said size of a black pepper, but still visible to the naked eye. And upon careful dissection, the plexus meshwork of these sympathetic division is also visible. So this is just to give you a little idea of how tremendously interconnected all these individual structures are. So where do we take nerve impulses through the sympathetic division? We take them to the sweat glands. I mentioned this in my example, erector pili, smooth muscle. Many blood vessels are innervated by sympathetic nerve impulses going through spinal nerves. From the thoracic and celiac, which is in the abdominal cavity, celiac plexuses, we have supplying nerve impulses to heart, lungs, some parts of our digestive system, and some blood vessels. And we have the so-called splanchnic nerves, which is lower abdominal or pelvic cavity organs. So some part of our digestive system, urinary and reproductive organs receive nerve impulses. The one of the curiosity and unique aspect of just this sympathetic division is that we can spread nerve impulse going through the sympathetic division very, very quickly. In the postganglionic, I'm sorry, let me just step back. Inside the ganglia of the sympathetic division, the signal from one preganglionic cell is spread onto many postganglionic cells. So it means that a single signal will be spread and shared widely. This is called divergence. And so the slightest indication that we need to activate sympathetic homeostatic maintenance activities in the body is spread through many, many organs. So this creates a mass activation. Multiple target organs are activated incredibly quickly in a matter of seconds. This ability comes very handy when we will be explaining the fight or flight response. And also we have this unique situation. We saw the artistic rendition of this nerve impulses from sympathetic division entering the adrenal gland. And so the neurotransmitters epinephrine, norepinephrine are released into the blood because we are releasing products into the blood. These are also called hormones, and in chapter 18, AMP2, we'll be looking at these hormones. Here we'll be mentioning their relevance to the fight or flight response in the second part of this lecture. So let's take a look at the parasympathetic division. We are sending, so this is the craniosacral division, right? So no wonder that we are using cranial nerves to move nerve impulses out of the brain. Oculomotor, and the authors of this textbook and some questions on your comprehensive final may assume that you have these cranial nerves pat down from the previous chapter, chapter 14. Oculomotor nerve is cranial nerve 3. Nerve impulses to muscles that change the shape of the lens or changes the size of the pupil go through the cranial nerve 3. Facial nerve, cranial nerve 7. Nerve impulses that trigger tear production from lacrimal glands or saliva production from salivary glands go through here. Cranial nerve 9. Nerve impulses that stimulate the parotid salivary gland go through this cranial nerve. And vagus nerve, cranial nerve 10. If you remember chapter 14, this is the craziest, wildest cranial nerve. It not only enters the thoracic cavity, it enters the abdominal and even the pelvic cavity. Very, very long and wide-ranging cranial nerve. So nerve impulses of the parasympathetic nature that goes to the heart, to the lung, various organs of the digestive system, and also reproductive system will be going through cranial nerve 10. We also have the sacral aspect. So we just looked at these 4 cranial nerves. This is a craniosacral division. So we do have the sacral aspect. They bundle together as the pelvic splanchnic nerves, and we are sending nerve impulses to colon, ureters, bladder, and some reproductive organs through the sacral portion of the parasympathetic division of our nervous system. So this was the gross anatomy of the parasympathetic division, and in the second part we'll take a closer look just how is this dual innervation manifesting itself when we are reacting to some kind of an alerting or danger sign in our environment. Functioning of many of these dually innervated organs will change in a matter of seconds to maintain homeostasis in a very different way. So I'm going to stop here, and we're going to pick up the activities of our autonomic nervous system in part 2.